where were you in the months leading up to taking this position? Where were you on the, I guess, continuum of believers around the influence or capabilities of artificial intelligence in antibody design at the time? Was there any degree of skepticism? Were you sold? Had you been exposed?
I had not been exposed to it much for antibodies. I'd heard about it being used for small molecule design, but not so much for human antibodies. And actually, that was something I needed to learn about. Uh, but the biologic platform turns out to be a really interesting, powerful one because what they do is basically mimic what the immune system does anyway. They do it using machine learning algorithms,
Mhm.
but it essentially mimics the way we form antibodies when we need to make antibodies, say, to a virus, or in this case, to tumor cells, or not to tumor cells, to interleukin 2 that will then drive the activity against the tumor cells. Um, so I thought that this technology made a lot of sense because they trained it on a data set of real proteins. So it wasn't imaginary stuff. They looked at the sequence of real proteins and real antibodies that bound to those proteins and the epitopes on them where they bound. And the machine was trained on that data set of millions of those interactions and learned. So if the antibody has a binding regions called CDRs, and those have their own amino acid sequence, the machine learns if there's a particular amino acid in a particular spot on a particular CDR, that's likely to bind to a protein sequence that looks like this on the target side or the epitope side. And it can do it in a much more formidable way than the human mind can do because it's looking at massive data sets and basically doing pattern recognition and predicting what could be done to change the sequence of an antibody to make it even more specific and even higher affinity for a given target epitope. So, like I said, this is what our bodies do. When we get sick and need to make antibodies, the immune system asks, do I already have an antibody that will kind of bind to that target, either because I was born with it as a germline antibody or it's one I've acquired throughout my years living in the environment and being exposed to viruses and bacteria and the like. And then that serves as a template that then gets rapidly optimized. So this is why COVID killed millions of people in the US and worldwide, because most of us didn't have a template antibody to SARS-CoV-2. We hadn't been exposed to a virus that looked like that. And, you know, maybe a jump species. And we weren't, as humans, we hadn't seen anything like that. So a lot of us
Mhm.
didn't have antibodies. That's why we needed vaccines to create those antibodies. But that's exactly what we do when we need to make antibodies. Is our immune system says, is there a template? And then can I quickly refine it through a process called somatic hypermutation, introduce lots of changes in the sequences and find something that in a very refined way binds to that particular target with really tight binding and is specific to that target. So that's what Biologic did. And in the case of IL-2, they wanted to land an antibody just on a really tiny patch of IL-2, which is already a small protein to begin with. It's 159 amino acids. So not a lot of real estate. And they wanted to dock just on one part or just land on one part of that real estate
Mhm.
with an antibody and do it with high affinity. So they're not the first to try this. Others had attempted it using more conventional antibody generation technologies and were not successful in creating antibodies that would land just on that epitope, but still leave other parts of IL-2 exposed. That epitope or that region is the part of the cytokine that binds with one of the receptor subunits called alpha or CD25. So they wanted to hit that, but leave the other parts of IL-2 that contact the beta and gamma receptor subunits exposed so that they could then interact with those receptor subunits that are on effector cells. Because the difference between IL-2's activities depends on where it's binding. IL-2 can be either an immune suppressor or an immune activator. And generally, when we're healthy, we have low levels of IL-2. It's going to generally act as an immune suppressor because at low levels, most of that will be bound by high affinity trimeric receptors, so three-part receptors that are on T regulatory cells. The T regulatory cells have as their sole purpose to suppress the immune system. That's their job. They're very good at that. And one Treg can suppress 10 effector cells. So mentioning the effector cells, they're the ones that can kill tumor cells or virally infected cells. These are things like CD8 T effectors and natural killer cells. They express a two-part receptor, a dimeric receptor that lacks the alpha subunit. So the thesis was, if you can create an antibody, binds to IL-2 and prevents that part of IL-2 from interacting with the third subunit of the trimer, then you'll keep it away from the T regulatory cells. You'll also keep it away from the vasculature, by the way, because the vasculature expresses that trimeric receptor and that drives the tox with IL-2. But you could retain the immune activating portion of what IL-2 can do and expand and activate the CD8 and NK cell populations. We've proven that now preclinically. So we've looked, in vitro with isolated human immune cells. We've looked in animal models, and we've seen that we can drive the expansion of the types of cells that can kill tumor cells, and we do not expand the T regulatory cells. In contrast, we actually reduce them, and we don't have any of the vascular leak or any of the swelling or cardiac issues associated with high-dose IL-2.
How much of this would you say the complexity, right, was directly at least partially attributable to the AI ML influence?
Mm-hmm.
Like, I was going to ask you, sort of to rank the value or advantages of having machine learning algorithms contributing to molecular design, whether they be the design itself, the accuracy of the design, the efficiency of the creation, the understanding perhaps of a mechanism. But as you describe this, and the complexity and the sort of the finite, to your point, real estate that you had to work with, I guess I'd hone the question in a little bit on that. And I mean, quite frankly, maybe all of the above. Like maybe AI and ML contributed to that degree of complexity, and also did it in an efficient and far more efficient way than the human mind could have. You tell me, like what's the AI influence on this specific application?
Yeah, it's not to say that this couldn't have been achieved someday using other technologies, but it hadn't been achieved. If you look just at the evidence, others who'd attempted it were not successful in drugging this particular epitope on IL-2. So empirically, it was a struggle. And the AI can just process so many data points so efficiently, and again, recognize patterns that by brute force or human insight, it might take a lot longer to achieve. So it doesn't mean it couldn't have been achieved some other way, but it hadn't been. And I think it would take a much longer time than it did for biologic. And we've moved very fast as a result. So I don't think that AI is the answer for every problem, but it seems to be the answer for this problem. It's gotten there certainly first, and I think best.
Yeah. You talked a little bit about the team that you've put together, and you're a relatively small team. Was everyone in all in and convinced already, or was there some degree of management finesse that needed to be applied to get the traditionally biologically thinking minds married to the tech, the ML AI sort of mindset?
I didn't encounter that so much. And again, I have a fantastic team. The difficulty is with a startup, you've got to convince somebody to go to a company that has no track record at all. So they're really banking on the right people being coalesced to advance this company forward, and having the financing to do that. So that's probably where I had to spend more of my time in the recruiting effort, was assuring them that this is, while it's a startup, there's a very clear path here. And a lot of the risk has been removed because we know IL-2 is a really active cytokine. It's just very toxic sometimes. And this is a chance to do something that nobody else has. And the other sort of concern that people often have in a company at this stage, and for us, is this is the only project. And yes, that's right. It is our only project. It also means we can focus and go really efficiently with it. But it's the kind of project that if it works, and it looks like it is, it will have applications in many different tumor types. We think this has the potential, like the checkpoint inhibitors, which are used in many different solid tumors and liquid tumors. Again, they're immune activating in a way. They take the brakes off of T cells, so to speak, through the PD-1 blockade or PDL1 blockade. And that's something that's proven to be very effective for many different tumor types and is now integrated into standard of care in all sorts of cancers. We could do that here too. And so convincing people of that, took some time and effort, but I think the people who are attracted to it are the ones who are willing to take that risk and see the potential to make a big difference clinically and to build a great company. Mm-hmm. So people self-select. But, I chose, in some cases, people I knew or knew of. In other cases, it was people I didn't know, but I really liked their backgrounds and how they approached the company and the problems we're trying to solve. Yeah. So our chief medical officer, for instance, gave IL-2 to many patients when he was at NIH and knows how it looks and how the responses manifest and how the toxicity manifests as well. So it's hard to find that expertise in every medical oncologist. And that's a big reason I chose him. Plus, he'd been at other small companies. And, I thought he would, because of his experience with immune engagers and antibodies, he was a great fit. So that's an example. Yeah. And he is a great fit. He's doing great work. And then our chief financial officer was somebody I worked with before at Genentech. And I knew he was a very skilled and capable executive. And the rest of the team, again, we select on that basis. Are they willing to take the risk? And do they want to build something? And all of them do.
Yeah. IL-2, problematic as it may have historically been, is also a pretty popular space, in addition to a cottage industry of startups focused there. You're rubbing elbows with Amgen and Merck and Sanofi, BioNTech. What's the, I guess, what's the strategy from the CEO suite in terms of doing business in a pool full of big sharks, right? Like I promised in the intro that we would address this, being the, little dog in the big dog park. I guess two-part question, like what's your general strategy around that, and why? Like, what's the why behind your belief, confidence that you can play in that space?
Right. So I wouldn't wade nonchalantly into an ocean full of big sharks. Yeah. I'm not that dumb. So, I had to believe, and we have to believe, that there's something very different about what we're doing. And there is. So yes, it's a crowded space. There have also been some big failures and billions of dollars spent trying to improve upon IL-2, and hasn't really advanced to very far because mechanistically there have been issues with some of the prior approaches. Mm-hmm. We're doing something completely unique in the class, and I think it will emerge as the best in the class as a result. So when I look at all the competitors, and there are several, big pharma, but also small biotech, international competitors, it is a crowded field in that sense. But I think we can win because the entirety of that competitive class is varying on the theme of IL-2 itself. They're altering IL-2 itself. Mm-hmm. Through various strategies. So that's been pegylation, albumination, fusing to CD25, fusing to anti-CD8 antibody, fusing to an anti-PD-1 antibody, masking with units that are proteolytically cleaved in the tumor, theoretically. So these are all ways of changing IL-2 itself. And those address some of the problems, and they sometimes have reduced toxicity. Some have activity too. But because they're not natural proteins, very often they're difficult to manufacture. They frequently have short half-lives. The immune system sees them as foreign objects and tries to clear them, and actually makes anti-drug antibodies frequently to things that have exotic attachments or non-human sequences. What we're doing is completely different. We're using native IL-2 So we're not altering IL-2 itself. We're using what the body makes or a carbon copy of that, which is recombinant human IL-2, proleukin, that looks just like what our body makes. We're using that and we're redirecting it with a human antibody, which also looks like what our body makes. So from that sense, the Aulos approach looks a lot more native or natural to the immune system because we're using human IL-2 and human antibody. And we're not changing IL-2, we're redirecting it. So as I mentioned, our antibody lands on IL-2, does so with high affinity and a low off rate. So once it grabs the IL-2, it keeps it bound. And that complex can then activate the cell types we wish to activate, which are the CD8 T effectors and the natural killer cells. And a portion of the T cells as well go on to become central memory cells. So memory is induced, which is also very important. And we're keeping IL-2 away from the vasculature and the Tregs. So we will improve both safety, we believe, and efficacy as a result of that. So it's not to criticize the other approaches that are being taken, but they're totally different. And we're unique in the field in that we're going at this with native IL-2 and a human antibody. And human antibodies are great drugs. We know that some of the biggest brands, whether it's Keytruda or Humira or what have you, are based on human or humanized antibodies. So it's a tried and true and very effective way to drug targets in cancer and other disease states. And we don't see anti-drug antibodies. We see a long half-life in humans over 2 weeks. We don't see any of the safety issues that have been historically associated with high-dose IL-2. So it's looking very promising.
Yeah.
But I'm not afraid of the sharks.
I'd never guessed that you would be. All right, we need to wrap things up here. So final question for you. I was going to look for a sort of a clinical update and next steps, which should be absolutely fresh in your mind. As I said, coming off of the ASCO event, I'm sure that you've delivered some of the facts, figures, and stats repeatedly over the course of the last several days. Share a few of them with us here. What did you present down there?
Yeah, we presented a poster at ASCO on Saturday, June 1st. In that poster, we had data from the entirety of phase 1, and then the very first few patients in phase 2, just the first 4 patients in phase 2. So in phase 1, we were testing a variety of antibody doses as well as IL-2 doses because we have decided it's more effective to sprinkle in a little bit of low dose proleukin or IL-2, very low dose. And that seems to activate the immune system and lead to responses more often. So in phase 1, we were tuning all of those dials and we settled on our antibody dose and we settled on our proleukin dose in phase 1. Now in phase 2, the only thing that we're changing is the schedule of proleukin. So we're testing whether just one dose is enough to kickstart the immune system or if we want to give it every 2 weeks alongside the antibody. So we'll answer that this year. What we saw in phase 1 is that the regimen is well tolerated. It has a very good safety profile. Most the AEs were grade 1 or grade 2. They quickly reversed. We have seen some AEs associated with immune activation, so cytokine release syndrome, and that was quickly reversible and controlled with steroids and didn't occur in very many patients. And we saw some transient lymphopenia, which is more of a lab finding. It doesn't mean we're killing the lymphocytes, but it's known with IL-2 that when you give IL-2, it causes trafficking of the lymphocytes out of the vasculature. So for a period of time, the lymphocyte count in the bloodstream drops. It's likely going up in the tissue. And then that rebounds, and there's no clinical effect of that, nothing negative. So we and the investigators are very impressed with the tolerability and safety profile of this regimen. And then on the activity side, even in phase 1, we've seen activity. So in phase 1, we have patients with deep and durable responses. We've had some on over a year and notable responses in melanoma. So a patient who had not responded to prior anti-PD-1 and anti-CTLA-4 therapy came onto our trial and is still on treatment, has a 48% reduction in his tumor. And we had a bladder cancer patient in phase 1 as well, who has a complete metabolic response by PET. So PET measures the metabolic activity of the tumor, and it's clear that the tumor looks dead on the PET scans versus baseline. That patient's on therapy about a year as well. And then even in phase 2, which is early, we just started it, but we have already the first patient treated has a response, a deep response, melanoma, minus 76%. And again, that patient had not responded to anti-PD-1, anti-CTLA-4. So we're impressed by what we're seeing. We've got patients with shrinkages in lung cancer as well, renal cell carcinoma, colorectal cancer. So it's early still, but very promising based on what we've seen to date. And this year we'll complete the phase 2 cohorts, or largely complete enrollment in RCC and melanoma, comparing that schedule where we give either one dose of IL-2 with our antibody or every 2-week dosing of IL-2 with our antibody. And later this year, we'll open up cohorts in non-small cell lung cancer, both with and without the checkpoint inhibitor avelumab, or Bavencio, which is a product from Merck KGaA in Germany. We announced in mid-May that we have a relationship with them now, whereby they're providing us with drug supply for use in our clinical trial. And we've seen preclinically that this combination is synergistic. We can clear established tumors when we give our antibody AU007 plus IL-2 plus anti-PD-L1, a surrogate that looks like avelumab. And we selected that antibody in particular because although there's a lot of checkpoint inhibitors that are approved, both anti-PD-1 and PD-L1, this is the only one that has what's called effector function. So it has a competent Fc domain that will engage the receptors on natural killer cells, meaning that if a cell overexpresses PD-L1 and the avelumab binds to that PD-L1, a natural killer cell will then bind to that and attack and kill the tumor cell. So we like that because one of the things that we do with our AU007 IL-2 regimen is boost not only T cells, but NK cells as well. And the NK cells are the ones that can kill via this mechanism called ADCC. And avelumab offers that potential as well as being a checkpoint inhibitor.
Very good. Follow the science, folks. Follow the data. If there's
do that.
No, I mean the storyline here from all the time we spent talking about, your career choices and trajectory through what you're doing at Aulos. I mean that's a common thread of the story, and I appreciate you sharing it with us. Very instructive. And I like talking to you, Aaron. You're a pleasure to talk to. I love
you too, Matt.
Yeah, you can you have an innate ability to convey the scientific story in a way that people like me who are non-scientific by training understand. So thanks for joining. Yeah, no, thanks for joining me. We'll continue to pay attention to Aulos. I'd love to have you back on the show at a later date, because I feel like I could talk to you for another 2 hours, but we just can't afford to do that right now.
Sure.
So thank you for joining me. I really
happy to join again. Thank you. Really appreciate the time today.